Okay. So before we start the lecture of today, as usual, I would like you to go to the Brightspace, and let's go to the content on today's lecture, 19, and there you will find a Woo clap, and let's go and answer this. And please, today I will really time it because there's a lot of material that we still need to cover. Okay, so let's go to this format. Let me know if you can see the Woo clap questions because now there's another window that opened here. If not, please say somebody says we don't. Okay, yes, we see it. Okay, awesome. Cool. So first question, and I will start putting the timer to see how quick you guys are gonna answer the questions. So this also will give me an information about how I will calculate the time for you in the exam. So how does the percentage of acrylamide in a gel affect protein separation? The lower acrylamide percentage are better for separating smaller protein, or the higher percentages are better for larger protein, or the higher percentage are better for smaller proteins, or the percentage is not really important. I got only one answer so far. That's already 45 seconds. Okay, guys. 16 people already answered. There's 30 people on the class today. 19. Okay. So just for the sake of the time, I'm gonna really stop at this like less than a minute, and I show you the other. Higher acrylamide percentage are good for separating larger protein. No. Be careful. Majority of you said the right answer, which is 67%. Great. For those who said higher acrylamide percentages are better for separating larger protein, the higher the percentage, the more difficult for the higher protein to pass through it. So that's not going to be correct answer. Here's the correct answer for you. Let's go next. Oh, on the other way around. What happens to a protein at its isoelectric point during the page? And again, let's count a minute. It denatures and form aggregates. It does not migrate because its netted charge is 0. It migrates to the cathode toward the negative charge, or migrate toward the anode, positive charge, or none of them. So these are the types of questions that you should know the definition of the isoelectric point. So if you didn't know what is the isoelectric point, it's going to be very difficult for you to answer this question. So we are already 45 seconds, 10 more seconds, and I will show you the answer. Now, I am giving you 5 answers most of the time. You would automatically know in the, so it's already 1 minute, you would automatically know that when there are more than 4, this question is going to be a little bit more difficult compared to the regular, like 4 choices only. So the answer is 90% Correct. Awesome. So that's great. That's great. There was only two questions. So it migrates to the cathode, and yes, this is correct somehow, but it's not at the isoelectric point. The protein would migrate toward the cathode or the anode given its charge, right? But when we are speaking about at its isoelectric point, this means it has to stop migrating because its net charge is zero. So this is pure biochemistry. Oops, oops, oops. Sorry. I hope you didn't see the answer. Which of the following stains is specific and is used for visualizing protein after SDS page? So we have discussed multiple types of stainings throughout the course. Ethidium bromide was one of them. Comasi blue is another one. Cyber green is the third one. Red, gel red, and none of the above. So which one of these is a specific and is used to visualize proteins after SDS page? And SDS page meaning it's sodium disulfide. So this is a denaturing type of shock. So I just got the message from my TA saying that you guys should have already updated your assignment number 3, the bonus point. Is Joe Werner here? I thought Joe every time asked me, so you already got the point that we discussed. Okay, so what is the answer? You guys, I tricked you. I promise I will not be that nasty in the exam, but I tricked you. The question was, which of the following stains is used to visualize protein after SDS phase? I simply inject that word there, specific. Comasi blue is not specific. It binds unspecifically to proteins. But yes, I fully respect your intelligence that yes, Comasi blue is used for the detection of the proteins. So that's great that 86% chose the right answer in terms of which stain is used for the proteins, but be careful. And this is like the multiple choice. Be careful in your future exams that one word can change something. Ethenium bromide is used for DNA, is not used for RNA, because ethenium bromide intercalates to the DNA, and cyber green is for RNA. Okay, guys. Next, oops. So, let's see. Which type of page maintains the native structure and function of protein? Native structure and function of protein. Be careful, there are two components. When you say native structure and function of the protein, meaning that the protein is still able to perform certain functions. And we are speaking about page. So is it the agarose? It's the denaturing page. It's the native page or non-reducing page. Is the SDS? Or no page allow preservation of protein function? So the question is very like direct. Is there a page that allows the native structure and function of a protein? If yes, which one is it? So we have 13 answers yet. I will count 10 more seconds to answer this. Okay. And the answer is native structure, native page. So for For those who answered correctly, the non-reducing page is a native page. So there is no beta-mercaptoethanol and no SDS. Yeah, sorry, no beta-mercaptoethanol. Okay, now for those who answered agarose gel electrophoresis, be careful because that we did not describe agarose gel electrophoresis with the protein. We described polyacrylamides, amides, and acrylamides. Obviously, it's not denaturing, but the other component, which most of the time in books and in references, you would say SDS page, most probably it's going to be denaturing because you add beta-mercaptoethanol. And for those who said no page allow preservation of protein function, yes, there is the native page on the non-reducing page, which allows the function of a protein to be studied. Okay, now it could be, so we answered that one. There's one more question. I'll just take this here and what determines the migration rate of molecules in the polyacrylamide? So the rate of the molecules that run in the polyacrylamide gel. Is it the temperature that you are running at? It's the molecular weight and the charge. It's the concentration of the acrylamide and the staining dye. Or it's the pH of the buffer, or all of that. Okay. 40 seconds. And the answer is molecular weight and charge. So in agarose, we discussed that if you run DNA, DNA is negatively charged, it's going to flee away from the negative toward the positive. And depending on the size, the smaller the molecule, the faster it runs. In proteins, it's the same thing. We are giving SDS. The SDS gives a negative charge for the protein. And then the higher the molecular weight, it runs slow. And the smaller the molecular weight, it runs fast. So it's molecular weight and charge. For those who said none of the above, all of the above, temperature, for example, that doesn't play a role. Now, the concentration of the dye doesn't change. The concentration of acrylamide change, or the percentage of acrylamide, but it's not the concentration of the staining dye. The staining dye is like Comasi blue, is like ethylene bromide, is like, in this case, in the page, we use Comasi blue or silver stain. And the pH could play a role, but it will not really change for only a category of the molecular weight. It play a role over all small and large proteins. Okay, so then I'm assuming there's one question that I probably passed. Let me just go to the end. Okay, okay. Okay, so let me do this. Did we answer this? Okay. You guys didn't answer, but I clicked it by mistake, so I didn't place it. But what is the role of the LOXP site in tissue-specific knockouts? So probably this is more than one time I showed you this question in a previous time. Is it to mark genes for random mutations? Or is it to insert a fluorescent tag for the genome? Or is it to facilitate a specific exon for recombination by recombinase in specific tissues? It's almost the same wording in the question and in the answer. It's to enhance the expression of a target gene or all of the above. So obviously, this is this type of question you will have in the exam for sure. Okay, so now we answered all those questions. I hope this gives you a good idea about how the exam would look like, and we will continue in the next two lectures as well. So stop this guy, and we go to the presentation now. Okay, so today's lecture is the fourth lecture about discussing the protein analysis. The protein analysis, before going to that, please note that you have already received an email about the evaluation of the course that will be closing on the 29th of November. If you haven't done, please go and fill the evaluation. It's 3 simple questions. It's going to take you less than 2 minutes to fill. And it's your right to communicate back. This is an anonymous, confidential between you and the department. So you have your full right to evaluate the course. Say what you didn't find adequate in this course or what should be done to make this course better. I already mentioned that we need some sort of application, experimental application for this type of course, because a lab would be amazing if we could do these basic techniques and you guys would be able to understand it better. What I try to do here is to provide you with videos. I don't know if you guys are watching that, but that's the only way. So please come back to this course, to this slide, and you can click on these links and evaluate. I welcome your evaluation. If it's positive or negative, I will look at it later on in January after I receive from the department. So I'll appreciate any feedback you will send. So as for today, we are discussing the immunoblotting, immunoprecipitation, and flow cytometry, or fluorescence-activated cell sorting. So in the previous 2 lectures, we discussed about how we generate antibodies, how we use antibodies in ELISA. And I mentioned to you multiple times that most of the molecular biology techniques we utilize for studying proteins are based on the fact that a specific antibody or a primary antibody binds to a specific sequence or epitope of a protein. And then we will be able to detect that primary antibody with another antibody that is conjugated with whatever type of labeling. In this slide, it is an enzyme that converts a substrate to a detectable end product. In all these techniques, we will use the same method in order to detect those enzyme antibodies. And we discussed the page, we discussed how we make it. I clearly see some people didn't go through the material, so be careful. Like, it's very clearly mentioned how we make the page here. And we never mentioned anything about agarose when we discussed the proteins. So please don't be confused with what we discussed with DNA versus the proteins. Also, I noticed that the YouTube that I gave you, it was in the first session that we discussed the proteins. So I'm posting it back here for those who are still behind. So here you can see the percentages. It's clear in the table. The higher the percentage, the more resolution you can get for the protein. So this was one of the questions that I asked you in the workshop. Now, the percentage of the page also dictates how fast a protein can run. So bigger proteins cannot run the same speed with moderate size and smaller proteins. So that's how the separation takes place. And we stopped at this slide in the previous lecture where we mentioned that if we run a page, so we have a gel here of acrylamide and we loaded our proteins, different samples of proteins, and then we have a page of gel. we can either take that gel and we stain it with Comasi blue or silver stain, and silver stain is more sensitive. For each of those has different applications. Or we can transfer those proteins on the page into a membrane, a nitrocellulose membrane, which is like a piece of paper. One day you are in a lab, you directly see that this is just a piece of paper that allows the protein to be retained. And so we can transfer all those proteins into the membrane. And that's what we call the immunoblotting, because now, instead of having the proteins embedded in this gel after they run according to their molecular weight, I can have them blotted exact same way as they look like here, but now they are on the membrane. And then I can, for example, want to look on one specific protein. Let's say this is actin or a myosin or a surface protein, whatever that protein is. So I will have to find an antibody that is specific to that protein, which is the target of this study. And then I would incubate my antibody on the membrane for probably overnight or a few hours. And then the primary antibody will find the protein on the membrane. And then I would wash the membrane so that the antibodies that bind like unspecifically will be washed away. And then I would add the secondary antibody, and that secondary antibody has the detection method. In this condition here, it's again as an enzyme. When you provide the substrate to that enzyme, it will convert it to light. And then you can detect the signal, and you would say there's more proteins here and or less proteins in one situation compared to another. So how do we do the immunoblotting? So there are a couple of slides here that are mentioned in the previous lecture and in this lecture. I'm reassuring again about reducing and non-reducing page. Non-reducing page is also known as native. And that's why I wanted to stress on this point. If you read it somewhere, the non-reducing condition, it's the condition which allows you to detect protein in its native state. And the most used reducing reagent is the beta-mercaptoethanol or the 2-mercaptoethanol, because that's the one that destroys the disulfide bridges. So if you got an example here where you have no beta-mercaptoethanol or beta-mercaptoethanol on the sample. So you take one protein sample and you added beta-mercaptoethanol here, and you take the same sample, but you didn't add beta-mercaptoethanol or 2-mercaptoethanol. And this is a representation how the protein looks like. So this is a protein that is bound by a disulfide bridge. So when you don't have the beta-mercaptoethanol, then the protein stays in its secondary structure. In other words, the two components are together, and then it shows a 80 kilodalton molecular weight. Now, if you add the beta-mercaptoethanol, this protein will be broken into two parts, and you can see that this part is smaller than that part. Then that's the big part here, and that's the smaller component there. So with the beta-mercaptoethanol, you will see two components of that protein on the page. So if you are looking to check the secondary structure, it's better to use the non-reducing or native reagent. And here you can also see that there is a protein that is made of two different proteins. One is 80 kilodalton and the other is 40 kilodalton. In this situation, again, if you use beta-mercaptoethanol or not, you will break it into either these two components or that component. The idea is clear. It's like just mentioned 2 times on the gel. Then if you are studying the native structure, like you want to keep the tertiary structure or the quaternary structure, so definitely you have to use the native gel, the native page, because that preserves the protein-protein interactions as they happen in the cell in vivo, and you can recover the activity of the protein, because if this is an enzyme, it's active as it is like this at the quaternary structure. If you make it in a reducing reagent, it will go back to the primary structure, and the primary structure, that enzyme is not going to be active. So therefore, it allows to recover proteins in their functional form, such as enzymes. So this was also the question that I mentioned in the MOOC lab. Now, in order to do the immunoblotting, we first have to do the SDS page. So we do only one step extra to perform the Western blot or the immunoblotting. So we start by taking whatever sample, it's blood or cells or tissues. We're going to extract the proteins from that, and the total proteins are loaded into the SDS page. In each sample is loaded in one well. So for example, we have here 1, 2, 3, 4, 5, up to 10. The first 4 could be normal cells. The other 4 could be tumor cells or treated with a drug or whatever. And then you take all the lysates from those cells. You take similar amount, let's say 20 microgram, 30 microgram. You calculate how much the volume, and you loaded the same concentration or the same amount of protein here. And then you run. We discussed that we have a marker or molecular weight that we know. So we know the molecular weight of this blue, this red, this blue in the first. And this is our reference to estimate what is the molecular weight of the protein. And after running, we will have the samples. We don't see them like this unless we do the ponso, which is there. Oops, escape. The ponso, unless you do the ponso like that. Okay. So the next step is to take this gel where the proteins are within the gel, and you will put it in a transfer setup, and that transfer setup has paper around to protect the gel and the membrane, which is stuck directly to the gel. And then you apply electric current the same way you apply it from up to down, but this time you apply it within the surfaces, from one surface to another. In this way, it's applied this way. If you do it for 1 hour, all the proteins within the gel, they migrate out and they find the membrane just in their face, so they're stuck to that membrane. So you end up having a copy paste of that gel with proteins all over, and it would look like the ponso here. So you have the molecular markers now. Instead of being on the gel, it will be on a membrane. So this is a piece of nitrocellulose paper. It's very thin, it's very delicate, and you have all the proteins. So each sample is indicated in those lanes. This researcher had a marker in the beginning and marker at the end, and it will retain the same size and the same band. So if you have a band here, it will be exactly the same band. Now, I have the proteins on the membrane, and this membrane is going to be taken to be stained with the first antibody for our protein of interest, and then the secondary antibody to detect the primary antibody. So I'm giving you a couple of examples of how we visualize the Western blot. And you will see in every publication you will read and every research paper in molecular biology, there should be either qPCR or Western blotting. So these two techniques, you should really, really know how they work. So the immunoblotting, or the other word naming, is the Western blot. So you can detect and examine proteins of different sizes and amounts. So you can, when you say different sizes and different amounts, I can go back again to this ponso. So you can tell automatically that this is a higher molecular weight protein, and that's a lower molecular weight protein, because this is around 250 kilodalton and that's around 25 kilodalton. So the running is like that. This is higher molecular weight, this is moderate molecular weight, and that's lower molecular weight. So we can differentiate the sizes already. The similar way we differentiate the sizes with the Comasi blue and silver stain. But now we have them on a membrane. Here we can stain them with antibody, but there we can't stain them with antibody. Now, the next thing we can, yes.
Do you mind just putting on the red pointer so when you move your
Oh, okay.
you can see it better?
Sorry, yeah, sorry.
No, thank you.
I'm gonna go back. I'm gonna go back to explain this because I missed the pointer. Thanks for letting me know, because when I go back and forth, the slides are freezing and I had to go out of the pointer. So what I was saying is that the Kumasi blue or the silver stain are pages, and all you do is just that last step where you take a membrane similar size to the page, and you put it side by side to that membrane, and you put paper around the membrane and the gel, and you apply electric current so that the proteins that are embedded within these membranes, this or that, let's take this an example, then they will migrate out of that gel, and since the membrane is not allowing the protein to pass, they will stick to the membrane. Usually we load the marker in the first lane. This researcher here applied the marker in the first or the second and the last lane. When I say the Western blot allowed you to check the results in terms of the size and concentration of proteins, because this is usually the way it runs from up to down, right? That's where the higher molecular weights are, and we know the molecular weights of these guys. I know them by heart now after years of Western blotting. So this is like 250 kilodalton, 220, 100, 75, something like that. And then as you go down, this is the lower molecular weight up to like 10 kilodaltons of proteins. So you already know when you blotted your proteins from the gel to the membrane, you already know which one is higher molecular weight and lower molecular weight. Now, the second thing you can do is the Ponzo is not specific. So it's just showing the total amount of proteins, and it's unspecific to one type of proteins. But when you want to detect a specific protein on that membrane, you have to do this. You have to know which antibody you have to use. Like if you are looking for actin, you have to have an antibody against actin. And that antibody will detect only actin out of all the proteins you can see here. And then you go to the classical way of detecting the primary antibody with the secondary antibody, whether fluorescent or whether by enzymatic change of the substrate. So if we look at this, it's telling you that the polyacrylamide is the first step where you isolate proteins, run them on the gel in similar amounts, and you have them in the gel, you transfer them, and now you have the membrane ready. So there is a detail that I added here for you, that normally the SDS page has a stacking upper part and a resolving lower part. I'm not going to ask you about this in the exam, but this is something when you go in labs, you should know how to prepare the SDS. In the protocol, it will tell you how you make this and how you make that. So you have to be careful that resolving should be down in this part. And the stacking is somewhere on the top. Most of the time we throw it away before this step. So just keep this in mind. There is a very nice video here to show you how the Western blot and the SDS page are made. So at this point, we have our protein of interest somewhere on the membrane. Again, many times before doing the experiment, we should know an idea about what we are doing. So if I'm working on a protein, let's say it's periostin, I should know the molecular weight of the periostin, or the SMAD2 and SMAD3 that you saw in the assignment. I should already know what's the molecular weight, and I should know the antibody binds to which epitope on those proteins, stuff like that. So I have an estimate on where those proteins should look like. Is it like in the 20 kilodalton area, or it's like in the 50 kilodalton area, or it's like 100 kilodalton area, right? So it's important to have that. Now, I was on this slide before going, and your colleague told me about the pointer. So we can examine the changes in the protein size and amount. So here's an example. How can we differentiate between conditions where a certain protein is changed by Western blotting? So the example is giving you here a neuron and microglia, two different types of cells in the brain, okay? And I intentionally removed what is the name of the protein. We don't care what is the name of the protein. But it's this Western blot clearly tells you, so the researchers normally, when they present this in a paper, they cut out the molecular weight marker. I don't know why they do that, but most people do that. And then they post these numbers. So these are the references of the molecular weight. So that's the area where you can see 100 kilodalton. If this is, it's 75. If it's between these two, it's around 60 something. If it's between these two, it's around 30 something. So it's a correlation, not very accurate. But the point is, if I look at this Western blot, I can not only detect the size, which I already can tell that this molecular weight is higher than that, because that's at the level of 75, that's at the 7 of 60, that's as around 30, and so the molecular weights are going lower and lower. The other thing is, I can quickly tell that this protein does not exist in neurons, or very, very, very little in the neurons, while it's expressed very, very highly in the microglia. And if you look at this protein here, you can tell that this protein is lower expression than that protein in the microglia. So you can directly correlate saying this protein here is minimally expressed in neuron, but highly expressed in microglia. So probably it's specific to microglia and not to neurons. And if you look at this protein here, it's the inverse of that. So this protein is highly expressed in neurons and lower expressed in the microglia. So you can tell based on which antibody you used if the expression is higher or lower based on the conditions. And you can make any different scenario here you want. You can say these cells are not treated with something and they're treated with a drug, and that drug is inducing a protein expression. Or you can say the drug is eliminating or downregulating a protein expression. If you look at this, it's already like all the scenarios you can draw, it looks like that. Now, the intensity of these bands is not only by eye. This is a black and white. In this condition here, this is black and white, where there is almost no expression and there is a lot of expression here. But if you have a scenario like this guy here, you can't really get a conclusion. So what you need to do is to quantify the bands. You quantify the intensity of these bands in order to say, oh, this is like probably 30% compared to this protein. Now, how you do that, this is a whole different story. This is very easy done. I put a video for you here. In one day, if you go to a lab and you're doing a Western blot and you got already to this stage, you watch that video and it's very easy. You will create a region of interest around those in a program, and it will tell you the intensity in each of these, and it will directly give you a correlation of the concentration of the protein. Obviously, there is more of this protein expressed compared to that in these different cell types. So Western blots are laborious. You could face many, many difficulties to get a Western blot work. I assume you guys had already seen this picture from the figure 1 in my paper that I gave you in assignment number 3. In this Western blot, obviously I cut the Western blots, so there was a whole Western blot like this for SMAD2, another one for SMAD3, another to TGF-beta receptor 2, another one for the CRE, another one for GAPTH. So there were 5 like this, but then I cut them to make them look nice. around the bands of interest. And then in this one, for example, the antibody is against SMAD2. And you have the two conditions. Recall, and I'm trying to explain this to you because this is important for your assignment. The idea is I had floxed SMAD2. And so around some exons in SMAD2, there is a LOXP site. So if I express a CRE in cells, I should delete SMAD2. And that's exactly what you see. So if the CRE is expressed, you can see here in these three conditions, there is a CRE that is expressed, and there's no CRE in these three conditions here. I just used a virus that doesn't produce CRE. It produces a beta-gal gene. Okay? So if we read it from here, in these three conditions, there's no CRE, and in these three, we have a lot of the protein CRE. Now, in conditions where I have a SMAD2 LOXP, the CRE is going to go and delete the SMAD2. And that's what you see here. And if there is a LOXP around SMAD3, then you're going to delete SMAD3. And if there's a LOXP around TGF-beta receptor 2, then you are deleting TGF-beta receptor 2. And in order to show the community that I'm not really just showing, probably I loaded less proteins here. I show them that I loaded exact same amount of proteins in all the 6 conditions because that protein is not affected by the process of CRE or LOXP. So that's my reference gene. Okay, so this is one of the application of the Western blot. So we could detect that a gene of interest that we are deleting can be easily changed. Now, some of you might say that SMAD3 is not 100% deleted. I agree. Probably some of the SMAD3 stays there. So again, to sum up, you have an antibody here that detects CRE, antibody here that detects GAPTH, antibody that detects receptor 2 of TGF-beta, SMAD3 or SMAD2. And each of those was a separate membrane. Now, how Westerns are done, again, this is the YouTube. And I put you here in the future, say you are doing a Western blot, what are the troubleshooting that you should do? So obviously we have to isolate the proteins. So this step is important. We have to know what buffer to use. Then we are generating the page. So we have to know which percentage of the page we need to do based on the antibodies we're gonna use or the protein we are expecting. CRE is very small. It's like around 20 kilodaltons or so. You can't use a low percentage for that because it will run out. GAPTH is around 50. So any type of gel is okay. So these questions you should ask yourself before doing the experiment. Okay, so I'm just like now thinking with you loudly. So one day you need to come back to do this experiment. Then you transfer your page into the membrane. So you have the blot. You can see that each band you have here has been blotted. So you have 4 there. You have all the 4 blotted here. Exact copy paste. Now, instead of the proteins being here, all the proteins are on the membrane. And then we block. The blocking means it's just that the antibody doesn't bind anywhere. It just, if there's a site on the membrane that probably antibody unspecifically bind, we put a blocking. This could be BSA, bovine serum albumin, or milk. But that will not block the antibody. It will still allow the antibody to bind to its antigen. And then this is the band we are expecting to see, or this is the protein we are looking for. So that's the antibody specific to it. So that's the primary antibody. After the incubation, it binds to that. Then when we put the secondary antibody, it recognizes the first antibody. That's a very easy step. We already see it many times. And then in order to detect where is the first and secondary antibody bind, we add the substrate to change. Now here it's HRP, which horseradish peroxidase. Okay, and then after that, all we do is just take an image with a machine that allows us to detect this substrate. And you will end up having this result on each membrane. Okay. After that, you will do the quantifications. In this paper, we didn't really need to do quantification because the results were almost black and white. Okay. Now, with this, I will stop for a few seconds here, and I want to ask you if you have any questions about Western blot. For me, it's one of the most important techniques you guys really need to know. If you want to keep your questions after, I can't really stay long today because I have an appointment, but I can meet you up in the office hours or in the next lecture before. Okay. Let me see what, okay. So there was a couple of things. I have a question regarding the assignment. Okay. Joe, if you have could you please ask me this question later on? And I'm really sorry because today I can't. I have an appointment in downtown with my family doctor. But feel free, Joe, to send me the question by email, and I will extensively answer you back.
Would you rather me just join the office hours tomorrow? Because I was planning on coming anyways, so
Yeah,
I can just
yeah.
wait until tomorrow.
So one other thing I want to say, tomorrow the office hour might not be from 12 to 1 because there is an assembly for the professors in the department, and they decided to be between 12 and 1. So I will send you later tonight two possibilities for another office hours before
All
the
right,
end of
sounds
the
good.
week. Yeah, okay. So that's it for the Western blot. Then now detecting the Western blotting can tell us if there's a protein A or a protein B if we direct an antibody toward that, right? Now, if I want to detect if two proteins bind to each other, then I can also do it by a process called immunoprecipitation. And the idea is very simple. I could find an antibody that binds to protein A, and then I pull protein A down. If protein A binds to protein B, then most probably protein A will bring down with it protein B. Now, if A doesn't bind to B, when I pull down A or B, there will be no combination of the two. So that's how it's made. So the process is first find an antibody toward your protein in question. You don't necessarily have to know this protein. It's good if you know it, but you don't necessarily have to know what is that protein. Maybe you are just asking what proteins bind to protein A. Then you find an antibody that's specific to A, and you direct that one, and then you pull down. If B is binding, you will bring B with you. If B is not binding, then A comes alone. It's very simple. Now, in order to get to that point, we have to first do this step, and then we have to do a Western blot. So we have to get to a condition like this. We have to load your pull down material back to a gel, put the antibodies against A or B, and then detect if A or B are there. So let's describe the immunoprecipitation. So the antibodies are also used to purify an antigen present in a complex mixture of proteins, meaning more than one protein bind to each other. So we can imagine that this triangle binds to probably one of these proteins in blue. So all I have to do is to bring an antibody that is specific to that triangle and pull that down. In order to pull it down, I have to use something called agarose beads or magnetic beads, which allow those antibodies to bind. Yes, there was a question here. Very good answer, Gary. So your colleague was asking about the staining. Which one would we use? Silver staining is the most expensive and the most sensitive. Chromacy staining is less expensive and less sensitive. So Ponzo is quick because you can use it on the membranes. Okay, so as I said, let's imagine that this protein bound to its primary antibody. So in order to pull this antibody down, to pull it, you have to use something big, and that's a bead. And that bead has protein A or protein G, which in fact are antibodies captured to that bead. So what will happen is, first step is we take the protein of interest. We take the lysase from the tissue or the cells. Second step, let's say we have two conditions, normal versus tumor or cancerous cells. I will get lysase from here, lysase from there. I will put the primary antibody in both. I keep the antibody running around or shaking for a good period of time. The antibodies will bind or not to the protein. And then in order to pull down the antibodies that I added to that sample, I need the beads. So I add the beads. So the beads will capture all the antibodies here in orange. And then when I collect these beads, it's very simple. You centrifuge them and they will be pelleted. If you wash, this unspecific binding here is going to get away. Only the binding specific to that antibody stays. Then you do elution. The elution is by using the SDS and the beta-mercaptoethanol, and then you go to the Western blot. So this is a step before the Western blot. Now, what is protein A and G that are on the beads is in the next slide for your info if you want to learn what are those and how we use them. So in fact, your antibody is going to bind to a specific protein in the lysates. And then in order to capture that primary antibody, you need something to stick it to the beads. And this is either protein A or G. So the beads, when you buy them from the company, they come with either magnetic beads because you can pull them by magnet, or you can use saffrose or agarose beads, and those you can pull them by centrifugation. So the next step, when your antibody binds to that agarose bead and protein AG, when you wash, you get rid of any unspecific binding. You keep only the binding that is specific to that antibody. And then when you elute, you will have your protein of interest and your antibody. Now, if your protein of interest has another protein bound to it, you can detect it. Okay? So that's the co-immunoprecipitation technique. One more time. And there's a question, and then there's like a specific example for you here. So again, so you have many proteins. Some of them are complexes together, and some are not. So the protein in blue here is alone, where the proteins in yellow, green, and violet color are in complex. So if my antibody is detecting this protein here, there is a very good chance that if I put this antibody in the sample, it will not only bind to that protein, it will bind to that protein and pull down all the complex. So you can see here in the next step, when I added the antibody, it bind to this one, but indirectly it brought all the complex with it. So that's the antibody capturing step. Then in the third step is when my antibody has to be bound to the beads. It's not shown there, it's here. So your antibody and the complexes are bound to the beads. And then the third step is to do the elution. So now when you elute the proteins, you will have your antibody, you have the protein that you are targeting, which is the right antigen or the epitope you are targeting. And then at the same time, you pull down other proteins, the green, this larger one, and this yellow. So these are binding partners of your protein. So you can detect them. And here there's an example to differentiate between specific binding. So this is the protein in violet is bound to a protein in green. And when you capture the protein in violet, you automatically bring down with you the green protein. So this is a specific binding. When you do the washing and the elution, if you have unspecific binding, so for example, the green attached to the beads, when you wash it out, it will not be retained. So you can get rid of that if it's non-specific binding. Or if, for example, your antibody didn't pick any proteins, then also if you wash it out, this will be cut away. Only the proteins that are bound to the antibody will be detected. Now, the next step is to load those samples into the SDS page. Imagine you have here, this is one condition where cells were taken from normal cells, and you imagine another condition where cells were taken from cancerous cells. Maybe cancerous cells don't have that protein, or they don't have that protein, or they have another protein that bind to this guy. So you could simply detect what comes down after the immunoprecipitation with different conditions or with different drugs. And when we load these proteins, again, this part is a little bit difficult for you because you never did a Western blot. You load the whole lysates as an input. You have to load everything that is not bound. So when we take these guys out, everything that stays in blue here, we load it as a control, and then we load these guys there, and then we can detect if this protein is found or not. And now the antibodies you're using for immunoprecipitation, they could be used for the Western blotting as well. So you could use this antibody here, and you could use it on the membrane later on. In immunoblotting, capture antibody could be used for detection of MYC-IP. Here we are using protein MYC as an example. In the exam, if I ask you, the capture antibody could be used for immunoprecipitation and Western blot, the answer is yes. Okay. So this is kind of give you an information how you do immunoprecipitation. Obviously, this is really, really difficult for telling you like that. I hope one day in the lab you will be able to perform it. Now, there are other methods that we can detect protein binding. This, the BioID method, is based on the fact that probably proteins, let's take a protein in yellow here as an example. Before describing here, there's one thing I have to mention. In order to detect that this protein bind to those others, the binding has to be like strong enough. Because if these protein binding to each other and broken by the SDS or by the binding of the antibody or whatever, or if the binding is very short, then probably you can detect it. So this method, the BioID, gives advantage over the immunoprecipitation for that sense. So if this protein, we don't know which binding partner it has, then we can make a fusion protein with this guy. It's called promiscuous biotin ligase mutant BIR1, this guy. What this dye does is it adds biotin to proteins. Okay. So how can I use that advantage of adding a biotin toward another protein? In other words, wherever I make a fusion of these, my protein of interest and the BIR1A, then every time this BIRA adds a biotin to a protein, it is possible that my protein of interest is bound to that protein. because this way, these two proteins become very close to each other, attached to each other, and then that is the step which allows the BIRA to add the biotin to that protein. So simply after, if I take all the proteins and I look for the biotin-related protein, there is a very good chance that every biotin-related protein could be a binding partner to my protein of interest. So it's like, you know, it's like how the it's like a bait. It's a very simple, in very simple term, it's a bait. You attach the protein of interest with the BIRA, and you see where the biotin is added. Wherever the biotin is added, there's a chance that these two proteins are bind to each other. So there is also a YouTube about how this is done. So again, there's lots of details we have to look at. I'm just giving you the concept. If you read it somewhere, I'm giving you more references if you need it in the future. Not necessarily that you know every single step here. With this, we will move forward to the detection of the proteins in terms of the flow cytometry. Now, one last thing I missed to say, and you really need to know in this slide, it's the fundamental difference between the co-immunoprecipitation and the bio-ID. With the co-immunoprecipitation, you have to pull down a protein that is bound. Probably, as I said, there are chances that you don't get a result there. With the bio-ID, it's more sensitive in a sense that even if this binding was transient for a few minutes, then the binding protein will have a biotin, and therefore it will be more chances to detect that binding. So this is very important to know. Okay, little bit physics now. We're gonna switch the molecular biology toward physics to study a little bit about the fluorophores and what are the fluorophores and how we use them. And in the next lecture, as I said, probably it's going to be the review, and the last lecture would be the immunocytochemistry and immunohistochemistry. But this is important for you to understand how the fluorophore works and how we can use them in order to detect an antibody. So first of all, you have on top here the wavelength, and on the bottom you have the visible light range. You can see that our eyes are equipped to see between 400 nanometers and 700 nanometers from the violet toward the red, passing through the blue and the green and the yellow. Okay, so in terms of energy, the lower the frequency of a photon, the higher the energy. So you can see that the wavelength is inversely proportional to the energy. Okay? In other words, that's why the ultra UV light is more dangerous, because it has lots of energy compared to the UV light and the visible light. And the X-ray and the gamma rays are even more dangerous. Now, the higher the energy they have, the more capacity they have to enter our body. And the smaller, the bigger the frequency they have, the lower energy they have, and the lower capacity to enter our body. Okay, so why this is important? Because if we have a fluorophore that is in the violet area here, this means it carries more energy than a fluorophore that is in the green area or in the red area. And there's a big chance that this fluorophore can give energy or lose energy to become green and lose energy to become red. Okay, so these are basic physics. I'm not going to ask you in the exam about what is the difference between gamma ray and X-ray and UV light. But what I want you to know this, because we mentioned that we use antibodies that are tagged with a fluorescent tag, right? We mentioned like FITC or green fluorescent protein or YFP, which is red fluorescent protein. So for each fluorophore, there are two different patterns of wavelength that you should know. First, the excitation spectra, meaning at which wavelength you can excite that fluorophore to the maximum. And then if you excite that fluorophore, it will emit light, but it's not going to emit light on the same wavelength. It's going to emit light on a different wavelength. And the rule is, as you excite a fluorophore, it's going to emit light on a lower energy. In other words, you're coming from the ultraviolet green, violet blue toward the red. In other words, from the low wavelength toward the higher wavelength. So when you excite this particular fluorophore, it generates energy and emits light with a lower energy. So the difference between the excitation peak and the emission peak, that's the Stokes shift. And if you look at the amount of the energy that number one has in the blue, and the amount of energy you have in the three, the arrow indicates that it's lower energy. So you have a deficit in the energy. So the blue becomes green, I would say, not red in terms of color. So it loses energy and then emits light. So this is something we can detect in flow cytometers and in microscopes. And that's how exactly we use antibodies with fluorophores in order to detect certain proteins. And that's how we can make combinations. So here I'm showing you a combination. There's a very nice website here. You can go and play with those. So the first antibody is labeled with FITC, which is green fluorescent protein. And the second antibody is with PE, propylium iodide. And if I excite the FITC, so you can see here, if I excite the FITC with this laser, it's gonna emit light there. So if I want to detect FITC, I have to have a detector that detects this guy here. And if I want to excite the PE, I use the 561 laser, and it's gonna emit light a little bit further. So you can see the PE emission in there, PE excitation is there in this one. FITC excitation is here. FITC emission is there. So this is how the basics of the flow cytometry works. I can make a combination up to 5 or 6 different colors like that. The bottom line is every time you emit, energy is lost and the wavelength toward becomes higher. And this is the rule for all the fluorophores. Okay? Now, based on this, I want to go back one step to describe for you the FRET, or fluorescence resonance energy transfer. I can take a protein A and I clone with it a CFP or cyan fluorescent protein. So it's a blue color, so it has higher energy. Then I will take another protein B and I will have a YFP with it. So I will fuse it at the level of the cDNA. Remember, if you're making a fusion protein between CFP and A or YFP and B, you have with the start codon, you go all the way till the stop codon of YFP, you remove that stop codon. and then you insert the cDNA for A. So you have both proteins with no stop. The same here. Now, I have the CFP, it has a higher energy. And I have the YFP, it has a lower energy. Therefore, if A and B are not bound to each other, and I hit the CFP with a light, it will emit only blue color. But if the protein A and protein B are bound to each other, then the CFP and YFP are very close. And therefore, if I hit CFP with the laser, it will emit energy and it will excite YFP, and then I will get yellow color. So I will get a little bit blue and yellow. So first, I have to generate these fusion proteins. Second, then I have to express those fusion proteins in the same cells. And third, I will check if I excite the higher energy fluorophore, would it give me only the excitation emission for the blue, or would it give me little for the blue and a lot for the yellow? Because that indicates the protein A and B are bound together. So if no yellow is seen, one can assume that the proteins do not interact. If the yellow is detected, then I would say these proteins bind to each other. So that's the concept of the FRET. And if I want to give you an example here, this is from real experiments. The protein called calmodulin and the other protein called myosin light chain, they bind to each other. And their binding depends on the calcium. So if you stimulate with calcium, they bind to each other. So first, the EGFP is fused with the calmodulin. So whenever you see green GFP, you can see it's fused to calmodulin. And then whenever you see red, then it is the myosin light chain. So if I take these cells and start putting calcium excitation, or like stimulate with calcium, then you can start indeed that the green and the red bind to each other, and therefore the cells start to turn to yellow. The more you add calcium, the more this binding show up. And you can see here the cell is almost totally yellow. And this is a transient process, so it can go back to normal. Okay, so this is how we could detect protein binding by FRET. Two different fluorophores, and the binding exactly I just explained in the previous slide. Okay, I think I will stop here. I will describe the facts and the immunocytochemistry in the next last session. And I'm assuming next Thursday, I will just go to describe for you guys which slides are important and which slides are less important for your study. And I still have 2 minutes, so I will stop recording right now.
Excuse me, Professor, I have a question.
Yeah, just give me 1 second, Joe. I will just stop recording, and yes, you can ask
No problem.
whatever question you want. Yes.